We're at the 22nd Retrovirus Conference here in Seattle, Washington. We're with Ellen, and we're talking about K65R, and we'll talk more about what that means. And Ellen, can you kind of tell a little bit about yourself, your name, your full name, affiliation, and the work you're doing?
Yeah, so I'm Ellen White. I'm a medical statistician, and I work for the Medical Research Council Clinical Trials Unit, which is part of UCL in London. And I've been doing that for about a year and looking into HIV drug resistance.
Okay, great. The work that you do is about the codon mutations and so forth. It's important for people to understand what that is. So let's be a little bit helpful here and explain to them what this is and how meaningful it is.
So in a similar way to bacteria, bacteria can mutate against the effect of antibiotics, and HIV has that effect of the kind of so it can mutate against its normal kind of form and mutate against the drugs which were developed to fight the infection. And if a mutation develops in a particular place, then that can be highly detrimental to the ARTs that people are described, which in turn can affect their disease progression and virological failure.
So some drugs will be better with certain mutations and others will be better with others.
So the particular mutation that I've been looking at is the K65R mutation, which particularly affects Tenofovir, which, as I'm sure many people are aware, is the first the main drug, their first line regimen for HIV to fight against that. And with the K65R mutation, you're likely to have problems with that ART, with Tenofovir, if you have that mutation present in the HIV.
So the work that you do is to, we'll describe how you've done your research. And by the way, did you bring along the
oh no, I haven't. Well, I've got it in my bag.
Okay, well, we'll dispense with that, but let's just describe it. The poster session is a place where we all look at these big walls with posters on them, generally about maybe 3 foot high by about 5 foot wide. And some of them are paper, some of them are cloth, some of them are plastic. But they have your whole layout on it as far as what it is you did, and almost like mini, like PowerPoint presentations, serial down the line. But it includes a lot of drawings, a lot of lab work, and so forth.
And trying to get the work that you've done for the past few months to be as concise and as helpful as possible. Which,
and it has the objectives out front.
The bit of the background. So in this case, that we've been looking at subtypes and how this particular K65 mutation differs between subtypes. So for subtype C, which is the predominant subtype in sub-Saharan Africa, and comparing it to subtype B, which is mainly found in the US and in Western Europe, and that's been shown in lab experiments. So that's kind of our background, that K65R is seen to be more prevalent within those patients with subtype C. And then we kind of go on to looking at the analysis that we've conducted into that to back it up with patient data.
So the program that you did is including a lot of work that may have been accumulating, and you've learned from that, and now you apply it to your work. The background is what you use.
So it's seen, as I mentioned, it's been seen in lab experiments, but it's also been seen in patients in sub-Saharan Africa. So this K65R is seen to be high within those patients. So actually we wanted to look at how is this similar to, how do we see that within the patients within the UK? Is there still this difference between subtype C and B?
So that was your premise, that, that what you operated from, what you learned, and now you're investigating. Tell us about how your methods in which you...
So we took patients in the UK that have had a resistance test to look at how their drug mutations are within their HIV, and we, for those patients which we had a complete ART history, we looked to see whether, so it was a wide range of different regimens that patients were on, and we looked to see whether K65R was prevalent within those patients, looking by subtypes B and C. And what we found was, to back up these lab experiments and these other papers that have been have suggested that this is the case, that yes, it comparing all different regimens, subtype C still has a much higher risk of having K65R compared to subtype B.
Okay. And so the results, so you proposed that as discussion, or was it a result?
So that was our conclusion from that. But I suppose the main conclusion that the main discussion that we have from this is, yes, we see that K65R is more prevalent amongst subtype C, which is clearly worrying. As I've said, it's quite it could be, along with other possible mutations, detrimental to a patient's success. But we want to see what is that effect down the line. How does their disease progression, how is that affected by this mutation? And that's going to be the next step, as to compare if virological failure is more prevalent in subtype C.
But this next step that's where your research ended with the inclusion. So
we've looked preliminarily between these subtypes, and we found
that you now have a next step to move to in the next phase of research that you'll have to do.
Yeah, definitely, and that's going to be the next the next phase, as to how will this affect the patients? How will that affect their treatment outcomes?
Right. Clinical outcome is extremely important. Obviously, that's the whole thing. That's the ultimate goal of what you're working towards. So, when do you think you'll get started on this, or will you be involved with that?
Well, hopefully, as soon as we hope to kind of make sure that this information with the analysis that I presented at CROI is possibly published and released. But as soon as that, is underway in the next few months, then we hope to start working on looking at virological failure and treatment outcomes to see how these patients are affected.
I think you made the statement, this has been the most comprehensive look at K65R.
Yes, we think that that's the case. There's been a few looking at patients within the same population, because we're unsure as to, in sub-Saharan Africa, yes, we see a high proportion of K65R, but is that because patients have been failing for a long time? They don't have viral loads monitoring as much as we do in the UK or in Western Europe and the US. So could that be because of something else? So it's important to look at within the NHS and within patients that are under the same healthcare system, whether it differs in that sense. And we had 25% of the patients that we looked at had subtype C compared to their subtype B. So there is good numbers there to look at this difference.
Okay. Do you now? How did you? I guess I'm traveling in a different direction here, but the experience that you have here at CROI, was it welcoming to you? Did you feel like it was challenging, or how? Describe it.
Well, it's been great. As I say, I've only been working in the HIV research for a year, and this is my kind of first big conference, and it's my first time to America. So to get the opportunity to present the work that I've done, to be given a scholarship, a young investigator scholarship, which I think is incredible that they're allowing young investigators so early in their career to have the opportunity to just be a part of it and experience on this huge scale. It's just been, great.
this is the kind of thing I'm sure the IS and the CROI Secretariat likes to hear, because it is important. And I do know that there's a number of young investigators that were funded insufficiently to stay the full course of the conference. They came in and did their thing and had to leave, and they only brought something to the conference and not took anything away. So I lament that, but I think we need to, while a person's traveled here, you might as well stay an extra day or two. Take in the work that's being presented and take something away as well.
Well, yeah, because you get an experience of a different analysis that's been, learning about PrEP and PEP, and that's not necessarily part of my research, but to just get an opportunity to hear from the experts and the people that have devoted their career to that is really, it's pretty inspiring.
Let me ask you, how did you, specific areas, how did you feel about were you at the Young Investigators? Okay. How did you feel about that?
Yeah, it was great. I think it was a bit over my head, some of the kind of vaccines and the science possibly behind that. And I really enjoyed getting a broad experience on that, especially the community perspectives. And I think that would be great to have more of an opportunity to hear about more the history, because I've not been working in HIV research as the epidemic has developed, and that would be great to have an overview of how amazing the research has been and what people have overcome to get to this position. That would be my only comment. That would be great to hear more on that side of that.
I don't know if you had the opportunity to be there in the opening plenary where they had the Ngaliman lecture and the Bernard Fields Memorial Lecture, and also a tribute to Joop Lange.
Yeah, that was great.
I think I don't think maybe that gives you a sense of the how can I say it, the importance of every scientist and how important, and the loss of a scientist that was critical to our fight, how committed and how thoughtful we are about that person that we've lost, and remembering him and his wife and others that were lost in that airline disaster. So the experience overall has been good.
It's been great.
And you've learned something because you had a little more time to stay than what others do. And you also brought something to the conference, the K65R ultimate research. And I'm hoping that others will learn from what you've been able to bring here. And I appreciate the time you spend with us here on this set.
Thank you. It's been
great.
Thanks.
Thank you.